Hello everybody, we are going to start a new system which is the visual system. As you know, that visual system is one of the special senses, and the visual system is more interesting and diverse than other systems of the body. And also we use our visual system much more than the other special senses. Okay, so that's why visual system is more interesting and useful for the human. And as I mentioned before, that the visual system in other creatures could be much powerful than human, and some creatures they have less powerful visual system. For example, the butterflies, their visual perception is higher than the human. We talked about that before. And for example, the bats, they use their eyes less, so their visual function is less powerful than the human, right? And if you just think about the visual system has some limitations. For example, in frontal-eyed creatures like humans, we have limited visual field. That means we can see only the front of us, but not behind us. But auditory system does not have that limitation. With your auditory system, you can hear the sound coming from any direction, right? So that is one limitation of the visual system. Another thing is interesting. If you close your eyes, you can shut down everything, like you can stop any visual stimuli enter into your eyes, so you can completely avoid the sight or vision just by closing your eyes. But you cannot do that in your auditory system. You don't have any structure to close the ears. Okay, the important submodalities of vision are color, is very interesting one, right? We see the things in many different colors, although some creatures can see more colors than the human. Some creatures see less colors than the human. Another is the shape, also the size, right? The texture, motion, distance or depth, all those are the submodalities of vision. So with your visual system, you can see the things in color, many different colors, right? Beautiful colors. Then you can see the shape of the things, size, texture, right? 3D or depth or distances of different objects, right? Which is very important for our navigation or movement. And your retina captures all these things and send them separately towards the brain, and at different structures of the brain or visual part of the brain, all these things are processed and then combined together to give you the picture of the entire object. Visual field is the area we can see with our both eyes together. Okay, so first we'll talk about the visual field, then we'll talk about the peripheral organs of the visual system, that means the eyes. We'll talk about the anatomy of the eyes. Okay, then we'll talk about the extraocular muscles. Extraocular muscles help to move the eye. Okay, so we move our eyes with the help of the extraocular muscles. Then we'll talk about the regulation of light, how the light entrance is controlled or regulated, and two structures, the iris and pupil. Iris is a muscular structure that regulates the size of the pupil, okay, and by doing that the iris controls the amount of light entrance, okay. We'll talk about that. Then we'll talk about the lens, how the light is focused on the retina, the back of the eyeball. So if you see the eyeball, all the way in the back you have the structure, the most important structure of the eye, that is called the retina. Why it is the most important? Because the photoreceptors for vision, rods and cones, are located in the retina. Also some other important cells are also located in the retina. So we'll talk about the retina, okay. We'll talk about different types of cells in the retina, the layers in the retina, and the functions. We will talk about the photoreceptors, rods and cones, two types of photoreceptors, and their functions. And we'll talk about the visual pathway, how the signal from the retina is taken to the primary visual cortex, okay, all the way to the brain. So we'll talk about those things first in the first part of the lecture. So you know that human vision is binocular vision. That means we use our two eyes with overlapping view, and that allows a good perception of depth, okay. You know that with one eye, although you can see the things, however you need two eyes to do the depth perception, especially fine depth perception. If I ask you to put a thread in a needle, you will need two eyes to see the hole in the needle and the thread. So with one eye you will not be able to do that. So that fine depth perception needs your two eyes work together, okay. That is the binocular vision. And your left and right eyes are located apart. How much? In adult head, the right and left eyes are separated by about 6 centimeter distance, okay, or apart. That means what? That means you see the same object in a space in slightly different locations, because your eyes work as camera. And now you just think that if you put two cameras slightly apart, that means slightly different locations, and take the picture of something, then what will happen? The picture will not be exactly same, right? Will be slightly different, and that information is called the binocular disparity. Your right and left eyes see the things in a space in slightly different locations. Makes sense? Because eyes are apart horizontally about 6 centimeter, and that information your brain requires or needs to give you the fine depth perception, or to see the things better as 3D, okay, which is very interesting. Anyway, so our eyes, as I mentioned, are located in the front of the head. However, you know that there are many creatures, their eyes are temporally located, temporal-eyed creatures. Let's talk about the visual field. So in human, the total area of visual field is 190 degree in horizontal plane, That means sideways we see 190 degree. That's the entire visual field. Okay. However, with both eyes you see 110 degree, and with each eyes separately you see additional 40 degree. So 40 degree with your right eye you see, but you don't see this 40 degree area with your left eye. You don't see, only you see this much additional with your right. And in left side same 40 degree you see only with your left eye. This part you don't see with your right eye. Make sense? So now this entire part in the middle with green color, that is 110 degree, you can see with both eyes. Okay, that is your binocular vision. Okay, which is pretty wide. Make sense? So out of 190 degree, which is your entire horizontal visual field, 110 degree is your binocular vision, visible to both eyes. Okay, and that is important for your depth perception. Okay, as I mentioned earlier. Now if you see the vertical field, how much you see above and below with your both eyes, vertical field is total vertical field is only much less than the horizontal field. The vertical field is only 140 degree. That means 60 degree above and 80 degree below. So this is the straight front, right? And with your two eyes you can see 60 degree above and 80 degree below. So you can see about 20 degree more downwards, right? So that is your entire vertical visual field. Okay, so 140 degree. However, if you see a creature that has temporal eyes, that means eyes are lateral, laterally, positioned in your in the head, although their entire visual field is very large. So you can see the blue area. This is the entire visual field, right? Much wider than the human. However, the binocular visual field is much smaller than the human. Okay, so they can see any fly or prey, even in the back part of the head, like here, but it is difficult for them to measure the depth or distance because this entire area is outside of the binocular visual field, but they can see if something is flying by motion. So they depends on the motion a lot to capture the prey than the human, which makes sense, right? However, you know that by moving your head or by moving your eyes, you can change the visual field easily, which is an additional advantage. Okay, so now we'll talk about the optic array. Optic array is the entire visual scene that you see. That means from the objects, the lights are entering into your both eyes. So that is the optic array. So the special pattern of brightness and color. So the light enters from everything within your visual field, right? At different brightnesses and different colors. and that light enters into your eyes, both eyes, from different locations in a scene. Actually that is the optic array. Okay, and the brightness actually depends on the amount of light, right? If the amount of light is more, it is more bright. So brightness depends on the amount of light that enters into your eyes from the object, the reflected light. And the amount of light is actually depends on the number of photons in the light. So how many photons are in that light, and the color depends on the wavelength of the light. So as you know, the light enters into the eyes in different wavelengths. Different colors have different wavelengths, we know that, right? So the color depends on the wavelengths. For example, if the wavelength is 400 nanometer, that is blue or violet. If it is longer, the wavelength, like about 700 nanometer, this is okay. So here you can see that this person is sitting on a chair, and from all different objects within his or her visual field, light is entering right into his eyes from everything. And one thing, just notice that if something is behind another object, that part is invisible, right? So that is not the part of optic array. So not everything within the visual field is optic array. The hidden structures, from the hidden structures, light is not entering into the eyes. So those are not the parts of the optic array. So in this, when the person is sitting on a chair and looking forward, the sight will be like this. So you see, few things are not visible. For example, the part behind this lamp is not visible, right? So the light cannot enter into the eyes from that part of those objects. Okay, so those are not the optic array. Now when the person is standing, the sight is different slightly. Make sense? So you can see here, now he can see like, you know, this upper part of this frame, because she's standing and she can see this part, but before that part was blocked, right? So some areas are now are not belonging the optic array. So optic array changes with the change of the position of the eyes. Make sense? Or body. So in simple way, optic array is the structures from where the light is entering into the eyes and what the person sees. And as I mentioned that the brightness and color, those are two things. Brightness depends on the number of photons in the light, which is the amount of the light, and the color depends on the electromagnetic wavelength. Okay, now before we move to next slide, as you know, the stimulus for vision is the light. And what is light? Light is an electromagnetic energy. Okay, electromagnetic energy. And that electromagnetic energy has a broad spectrum. For example, the lower part of the spectrum is like AM or FM waves. We don't see that, right? But the TV uses these wavelengths, and the upper is for example X-ray, gamma waves. We don't see that, right? Our visual range is very narrow. It's like a thin band within the entire spectrum, and our upper part is the violet and lower is the red. Okay, and other colors are in between. As you move this way, that means the energy increases, the electromagnetic energy is more. Okay, however the wavelength is less. So as the wavelength gets smaller, the energy increases. Okay, energy is more, and this way the wavelength increases, that means the energy decreases or gets less. So short wavelength means more energy and long wavelength means less energy. Makes sense? So now if we just see this one, I can say that since red is the lower end, so this is long wavelengths, about 700 that I mentioned nanometer, and blue or violet is about 400 nanometer, and that is the narrow range of our vision. Above the violet is ultraviolet here, and you already know that some creatures like bees see the colors in ultraviolet, so they can see more color in a flower, and we don't see that. Okay, so now you got an idea about the visual stimulus and the visual field as well as the optic array. Now we'll talk about anatomy of the visual system. Okay, like all other sensory systems, in your visual system you also have two parts. The peripheral part includes two eyes, and the central part located within the brain and the spinal cord. Okay, so first we'll talk about the anatomy of the eye. Okay, so if you see the eye, an eye has the following structures: eyeball, eyebrow, eyelids, conjunctiva, lacrimal apparatus, extrinsic or extraocular eye muscles. Okay, and extrinsic eye muscles arise from a common tendinous ring. That means the ring of a tendon. So this is the ring formed by the tendons, and this is the eyeball. So from the tendon, the extraocular muscles arise and get attached to the eyeball. Okay, so that tendinous ring is located inside the orbital fossa in the skull. Okay, so that's how the muscles arise and get attached to the wall of the eyeball and help to move the eyes. Now your eye movement could be both voluntary and involuntary. You know that if I ask you to move your eyes, you can do that. Sometimes without your knowledge your eyes move, right? That is involuntary. Now when the eyes move, you know that if you follow something like moving from here to here, your both eyes move from this point to that point, right? And when both eyes move in same direction, that is called version. Okay, so version is the movement of both eyes in the same direction. And another term we use that is called the vergence. And vergence is moving two eyes in opposite direction. That means if I ask you to just focus on something with two eyes, then your this eye will move that way, this eye will move that way. That means opposite direction, right? But pointing on the same thing. To point on the same thing, you have to move towards each other. That means in opposite direction, right? This one is moving this way, this one is moving this way, and that is called the vergence. Okay, the paralysis or palsy of the extrinsic eye muscle can lead to problem or disorder in vergence that happen. Okay, so those are the structures of the eye, and the most important is the eyeball, right? Because eyeball captures the light or visual stimulus and sends to the brain, and then the brain processes. This is just a picture showing how the signal from the object is taken into the eye, and from the eye the retina captures the image. And as I mentioned earlier, retina does a number of things. Okay, when the picture of the object enters into the eye, your eye flips the picture vertically as well as horizontally. That means that if this is the top of the picture or image and this is the bottom, this part will go here on the retina and this bottom part will go to the top. Also same thing in right and left. The right side will go to the left side on the retina and left side will be placed or captured in the right side. So everything flips. That is one thing your eyes do. Also, eyes do what? Eyes makes the image very small, because just think that your retina is very small, right? But the things you see, for example, if you see an elephant, it's a big animal, right? So that big picture will be placed on the retina as a very small, because the retina, total size of the retina is very small, right? So on the retina, the picture is, the size is reduced significantly, right? So one is the eyes flip the picture and put the picture on the retina, reduce the size of the image and put the image on the retina. And also eyes break the image into many pieces, many tiny dots. Okay, it's like a puzzle. The kids play with the puzzle that has many pieces, right? Then they put them together to make the entire picture. So the picture captured by the retina is broken into many small pieces. Okay, and also separate the modalities or modalities. You remember I already talked about the modalities. So the color, texture, right, 3D, motion, all those information are separated in the retina. So you see that your eyes do all those functions, right? Flip, which is the inversion, upside down and sideways. Okay, then shrinks the entire picture significantly, makes smaller, break the image into many small pieces. It's like, you know, a big mail. You can you know, break into many small, you know, envelopes. So you can do that way. Your eyes separate them, and also you can compare this, like, you know, to the internet, www, you know, all different addresses, right, to break the things into different pages. Like in one college website you have so many things, right? So each page has its own address. So similarly, the retina will break into many small pieces. Okay, and also the modalities, or which are also called submodalities, are separated, and these information all are sent towards the brain separately, and they are processed separately in different areas or by different groups of neurons. You know, there are color sensitive neurons in the visual part of the brain, texture, you have motion sensitive and depth sensitive neurons, right? So also more direction sensitive neurons. All those different types of neurons are present in different structures in the visual part of the brain, and that's why they need to be processed separately. Then the brain has that enormous power to put them again together. All those pieces, all those, you know, submodalities, they are combined together to give you the entire picture and the size. So you see how beautifully the eyes and the brain work together. Okay, so from the retina, the signal goes through different structures. We'll talk about that, and taken to the back of the occipital lobe, where you have the primary visual cortex, and from the primary the signal is then taken to the other visual areas in the brain. There are more than 35 different visual areas in the brain, and you already know that visual part of the brain is a large portion of the cerebrum. So the most part of the occipital lobe, a large part of the parietal, and a large part of the temporal lobes are, you know, responsible for the processing of vision. So a huge portion of the brain is, you know, dedicated to process the visual signal. Okay, so now we will visual perception is the construction of a description of the external world based on the cues, as I mentioned, submodalities, the shape, size, color, right, elevation, depression, or you know, depth, motion, all those things help together to give you the visual perception. Okay. However, one thing is important here in case of visual perception. Of course, the shape, size, texture, color, right, depth, or edge, all those motion are important. However, for visual perception, another very important uh, thing is your past experience. The visual perception will be completely different if you don't have any previous experience about that object. If you never saw, you have never seen a bat, if I show you a bat, you will not be able to tell what is that creature, right? Or if you have never seen an owl, if I show, you will never be able to tell what is this creature is. So not only by depending on the shape, size, or other features you can perceive, your perception will be very different than a person who already knows or experienced what is that object is, right? So that is a very important thing for your visual perception. And the perception varies person to person, right? Each person has his own visual world and perception. Okay, so this is how the eye looks like from outside. So you don't see the entire eyeball from outside, only a small part of the eyeball you see, right? Most part is located inside the orbital fossa, inside, and we don't see that part. Okay, anyway, so here you can see those structures I mentioned, the parts of an eye: eyebrow, eyelid, eyelashes, right? Also you can see the iris. This part is the iris. You see the round colored structure, that is the iris, and it regulates the size of the pupil. Pupil is this dark round aperture or opening or hole. Okay, so pupil is a hole or opening inside the iris, and the size of this pupil is controlled by the muscles in the iris. And when you see a bright light, the pupil gets smaller. Constriction of pupil limits the entrance of the light. We know that. And in dark light or dark condition, the pupil gets wider. Okay. Also, parasympathetic stimulation constricts the pupil. Sorry, parasympathetic activation, which is a branch of the autonomic nervous system, will constrict the pupil. Okay, and sympathetic, which is another branch of autonomic, activated by fight or flight response, this dilates the pupil. Okay, and also in bright light, the pupil constrict, and in dark, pupil dilate. Okay, so just know these few things. So the size of the pupil changes. Okay, and if your motor function of your brain is not working, then pupil dilate. Okay, so when someone gets unconscious, you'll see that doctors check the size of the pupil. If the pupil is dilated, that means the brain is not sending motor signal to contract the muscle. Okay, so this white part around the iris is called the sclera. You all know that, I am sure. Sclera. Only a small part of the sclera you can see, which is just around the iris. Most part is inside. We don't see that. Okay. So through the pupil, the stimulus, the light, enters into the eye. Okay. Another thing you have here, that is the cornea. You see, like thin glass-like structure that covers the iris, that is the cornea. So this colored structure is the iris, but the covering of it, the outer covering by a thin, transparent glass-like structure, that is the cornea. The iris gives the color of the eye. As you know, that different people have different eye colors, and that depends on the pigments in the iris. Okay, and that pigment is the melanin, and just remember that when we see someone's eye color is green or blue, that is not directly given by the pigment. The reflected light changes the color of the iris. So actually the exact color of that green-eyed person's iris is not green or not blue. For the blue colored eye, the light that is reflected, that changes the color to green or blue. So we don't have green or blue pigments in the iris. Okay, so now we will see the entire eyeball from outside to inside. Okay, so if you see the eyeball, let me draw here. Okay, so you know that this is the cornea, and this white part, which is the outermost structure, that is called what? Sclera. And these two structures belong to the outermost layer of the eyeball. So cornea is the transparent part in the front, right? Highly transparent, and it allows the light enter into the eye without any blockage or any change. So highly transparent structure. And the cornea is actually made of a protein, and in cornea you have free nerve endings. So free nerve endings are located in the cornea and that's why if you touch the cornea, you see the reflex. The eyes get closed because it is highly sensitive because of the presence of the free nerve endings. Okay, and those nerves are unmyelinated nerve endings. Okay, so there is no myelin sheath, so the light will not be blocked at all. Unmyelinated nerve endings, and those nerve endings make the cornea sensitive to touch, temperature, or any chemical. Okay, um, there is no blood vessel in the cornea, so this is the cornea, only free unmyelinated nerve fibers, no blood vessels, so the light will not be blocked. Okay, and as I mentioned, touch would involuntarily close the eyes. That is the reflex that occurs because of the high sensitivity of the cornea. Okay, now the question is, if cornea doesn't have any blood supply, then how it gets nutrition and oxygen? The nutrition and oxygen is supplied to the cornea by the tear comes from the the lacrimal gland. It has nutrition and oxygen, and also behind the cornea you have the anterior chamber here, and this chamber has the fluid that is called aqueous humor. The fluid is located behind the cornea, and that also has nutrition and oxygen. So by diffusion from the tear and aqueous humor, the fluid, the cornea gets its nutrition and oxygen. Okay, the cornea is about half millimeter thick, so very thin, right? Glass like layer in front of the eye, and that allows the light easily pass through it and enter into the eye. When the light passes through the cornea, small refraction occurs. So this is the cornea. Okay, so when the light passes through the cornea, slight refraction, that means convergence, inwards bending of the light occurs. Okay, and when light passes through the lens, which is behind the cornea, then more refraction takes place. Okay, so the light will be focused on the retina here. So two structures refract the light. The cornea slightly refract, and then the lens, the real lens, refracts a lot to put the light or focus the light exactly on the retina. Now the cornea is known as fixed lens because the shape of the cornea does not change, it is fixed. So fixed lens of the eye is the cornea, and slight refraction occurs, and the actual lens, crystalline lens, is this one, and this lens changes the shape. Sometimes it gets flatter, more flat, sometimes more round, like this. Okay, so the actual lens, which is the crystalline lens, it changes its shape, and by changing its shape, it focuses the light exactly on the retina. That's why the lens is called the flexible lens. Make sense? So those are two types of lenses in the eye. Anyway, so we are talking about the outermost layer of the eyeball, which has two structures. Only the front part is highly transparent, which is the cornea, which forms about 1/6 part of the entire outer covering, and rest part is white, which is the sclera, opaque, does not allow any light pass through, and this part, sclera, is about 5/6 part of the outer surface. Okay, so these two structures together form the outermost layer, which is the outermost layer, which is also known as the fibrous layer. Okay, and then we'll see the middle layer. So outer layer is formed by cornea and sclera. Okay, then the middle layer is the vascular layer. Okay, so we'll now see the middle vascular layer. So this is the middle vascular layer. You see here. That means in this layer you have the blood vessels, right? So blood flows in this layer. Most part of middle vascular layer is called the choroid. So choroid is the most part of the vascular layer. It has the blood vessels. And then if you see the front part, in the front part, this vascular layer forms a couple of structures. Ciliary body. Remember, the ciliary body is the ciliary body, okay, is this part, muscular part, and then this layer continues, goes to the front like this, and forms another very important structure that you can see from outside. That round structure gives the color of the eye. This is the iris. So you see, these are three important structures formed by the middle layer. Choroid, most part, highly vascular, and then the ciliary body and the iris. Okay, and you know the whole inside the iris is the pupil. So choroid, ciliary body, and what? Iris. Okay, and then the innermost layer, which is actually located only in the posterior part, like this, the green one. I am showing with green. So this is the innermost, most important and sensitive layer. That is the sensory layer that forms the retina. So number 3, sensory layer only in the posterior part, and that is the retina. Why it is the most important? Because the photoreceptors and other important cells are located in the retina. Okay, so this is the retina, okay, that captures the image. Okay, so those are 3 layers. Now we'll see a couple more structures you will be able to see inside the eye. Let me pick a color, this one. Okay, here you have the lens. So this is the lens. Okay, and the lens is held there by the ligaments. Arise from the ciliary body. So see, from the ciliary body, the ligaments get attached to the lens, and those are called the zonular ligaments, also known as suspensory ligaments. Okay, same thing. So arise from the ciliary bodies and get attached to the lens. So this is the lens, a crystalline lens that changes the shape. Okay, now here you can see 2 chambers. This is the anterior chamber, which is much smaller, and this big chamber is the posterior chamber, and this posterior chamber is filled with a jelly-like fluid that is called the vitreous humor. Vitreous humor, it's like jelly-like fluid, and that gives the shape of the eyeball, right? The pressure inside the eye to maintain the shape of the eye. And also this pressure keeps the retina attached to the wall, so prevents the detachment of the retina. And retinal detachment is a clinical condition. Probably some of you have heard that retina gets detached from the wall. One reason is if the pressure decreases inside the posterior chamber, that may cause that. So the vitreous humor gives the shape, maintains the shape of the eyeball, as well as prevent the retinal detachment. This is the anterior chamber filled with another type of fluid. This fluid is very highly water-like fluid, so this is called the aqueous humor. Okay. In some places, some books, you will see they have divided inside the eyeball into 3 cavities or chambers: anterior, posterior, and vitreous. I would say that just know that there are 2 chambers: anterior chamber in front of the lens and posterior chamber behind the lens, and posterior chamber contains the vitreous humor. Okay, that will make it simple. Okay, so that is the structure of the eye. Okay, now If you see here in this picture, let me quickly go over. This is the cornea, right? Highly transparent. And this is the sclera. They belong to outer fibrous layer. And then this is the choroid. You see the blood vessels, the middle layer here. And this is the ciliary body. And zonular or suspensory ligaments go to the lens. So this is the lens. Okay. Where the main refraction or bending convergence take place. Okay. And then this is the anterior chamber filled with aqueous humor, posterior chamber filled with vitreous humor. Okay. And this is the iris, part of the middle layer, and this is the pupil, the aperture or hole. Okay. So that is the basic structure of the eye. Now, if you see the eyeball, few more structures I want to show you. So again, now I'm going to show you the ciliary body. Okay, so ciliary body, and this is ciliary body, and this is the lens, and the ligaments. One thing is interesting here. What is that? When the ciliary body contracts, the ligaments relax. Okay, so kind of opposite, right? So when the ciliary body contracts, the ligaments relax. Now just think that this is your lens. Okay, and these are the ligaments, keeping it like stretched from both, from around. Now if these ligaments relax, these ligaments, just think that, are pulling outwards. That means pulling the lens from outside and trying to keep it flat. Now when the ligaments relax, then what will happen? The lens will get more round because the pressure is no more on it. So the lens will get more round. Makes sense, right? If I pull outwards, then it will get more flat. If I release that pressure, pulling pressure, then it will get more bulged or round. And when the ciliary muscles or ciliary bodies relax, then what happens? They work like, spring, will pull the ligaments outwards. So when the muscles relax, ciliary muscles or bodies relax, then what happens? They like move that way, pull the muscle away. So that will pull the ligaments, right? Suspensory ligaments. So now if you pull the suspensory ligaments outwards, then the lens will get more flat, like thin. Makes sense? Will get more flat or thin. So that's how the ciliary bodies regulate the shape of the flexible lens or crystalline lens, right? Sometimes it gets more round, sometimes get more flat. Now why? Your lens the shape of the lens needs to be changed. Think that here. This is the object. Okay, so the light is getting in, right? From the object, the light is entering into the lens, and the lens does what I mentioned before, refract the light to focus on the exactly on the retina. Okay, so from the lens here to the focus point, on the retina, this distance is called the focal distance. Focal distance. Now let's go back to the lens. So this is the lens. I'm just drawing the lens, and this is the retina here. Okay, so this is the object. This object is far. Makes sense? So the light is passing through the lens and will be refracted or converged on the retina. Makes sense, right? Now if I move this object closer, like here, the size increases, right? You know, this distant object is smaller, looks smaller, but if you bring it or move it closer, it gets bigger. So now it got bigger because I have moved from here to here. So now the light is getting in this way, right? So this light needs more converging more than this one, because these are closer, right? So needs less convergence to put on the or focus on the retina. Now since these 2 points are further away from each other, they need more convergence or more bending to put on the retina. To do that, the shape of the retina, the shape of the lens should be changed, should be more round, because thick or round lens can refract or converge more. So let me write here, thick or more round lens converges more. Make sense? And as you move the object away, object gets smaller, then you need less convergence, and the lens will get flat. So the ciliary body will now, you know, relax. So the lens will get more flat. We have explained that. I have explained that before. So now thin, when the lens gets thin or more flat, that means less refraction. Makes sense? Okay, so that's why you have the ciliary bodies, and they are changing the shape of the lens all the time. Okay, as you see the object is moving, coming towards you, lens gets more round, and as you move the object away or the object moves away, the lens gets more flat. Okay, and that is happening all the time. The function of the lens is focusing the light exactly on the retina. If the lens fails to focus the light exactly on the retina, if it focuses before or after, that means you will not be able to see clearly. To see the things clearly, the light must be focused exactly on the retina. Okay. There are clinical conditions where the lens loses its elasticity or flexibility, and that causes the refractory disorders, right? So let's stop this part now, then we'll start from the next slide in next part of the lecture. Okay, so in this part you have learned a few things. The stimulus for vision, then you have learned about the optic array, the visual field, right, and the structure of the eyeball and how the lens changes the shape to focus the light on the retina. Okay,